11. Dobrze. Kuzangpola, welcome to Botany Learning. My name is Spaldanyama, and this is a biology lesson for Key Stage 5, classes 11 and 12. Today, in my lesson, I'm going to talk about biomolecules. Before we look into the definition of biomolecules, I would like to first spell out the objectives of my lesson. By the end of the lesson, I will expect you to name the different types of biomolecules, explain the different types of biomolecules, and describe the structure of nucleic acids, namely DNA and RNA. All right, students, why is it that we have to know about biomolecules? Would you like to think for a second? Okay, biomolecules are important because it affects us every second of our life. Take a look around and observe the living beings or living matters. This is mainly composed of 6 elements, namely carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphorus. With the chief elements like carbon, hydrogen, and oxygen, along with other elements, this makes up what is known as the biomolecules, the building block molecules of all the living beings or all the living matters. This said, biomolecules play indispensable part in our life process, starting from their role in the disease detection to the proper functioning of biological processes in the lives of every organisms. These biomolecules are the compounds which contains carbon, hydrogen, and oxygen. These are usually made up of a smaller subunits called as monomers. And these monomers chemically combine to give rise to complex large molecules or subunits called as the polymers. For our easy understanding of what monomers and polymers are, I am going to give a simple analogy on the board. May I have your attention on the whiteboard now? All right, students. What I've drawn on the whiteboard is a chain. I've talked about monomers and polymers. Now I would like to make you understand what monomers and polymers are. You can think of monomers as a single chain in this entire chain. Now when these monomers combine together or they link up, they form a large molecule or a chain, which we call it as the polymers. So the simple analogy over here is the single chains can be thought of as the monomers, whereas the entire chain can be thought of as the polymers. I hope with this succinct introduction of biomolecules, you have learned an important aspects of biomolecules, or in other words, it might have helped glean some insights on the importance of biomolecules. Let's look into the definition of biomolecules. Biomolecules are defined as the carbon compounds, which comprises of carbohydrates, proteins, lipids, and nucleic acids that are found in living organisms or their cells. They are also called as the biological molecules. The first type of biomolecules that I'm going to talk about is the carbohydrate. I'm sure you must have heard this word so many times, but have you ever given a thought as to how these carbohydrates affect us? Before we try to answer this question, we're going to look into the composition, the definition, and the general function of carbohydrates. In the later stages of the slides, we're also going to look into the different types of carbohydrates based on their functions, based on their sugar units, and based on the number of carbon atoms present in the hydrocarbon chains. For now, please rivet your attention on the screen. Carbohydrates are compounds of carbon, hydrogen, and oxygen. They can be represented with a general formula, which I'm going to write it on the board. CnH2O N, where N is the number of carbon atoms, which is why these are called as the hydrates of carbon. And if you look at the ratio of the hydrogen and the oxygen, they occur in 2:1 form. They are also called as the saccharides, principally because their main components are sugars. The sugars make up the carbohydrates, which is why they're called as the saccharides. The simple examples of carbohydrates are sugars, glycogen, and starch. I picked up these examples with the hope that you would be able to remember, because these examples are quite common in our everyday life. All in all, carbohydrates are the principal source of energy. With this, I move on to the classification of carbohydrates. Based on the number of sugar units, we are going to classify or categorize carbohydrates under 3 subheadings. The first one is monosaccharides. The second one is oligosaccharides. And the third one is the polysaccharides. Let's try to define what monosaccharides are. For those of you who have entered class 11, these things would appear very new. For those of you who are already in class 12, this would be quite familiar to you. Anyway, for those of you who are enrolled in class 11, let me try to define what this classification of carbohydrates are. Mono, as the name suggests, means one. So saccharides means sugar. If you split up this word into two, it's going to come out as one sugar. Let us put up this phrase in a proper manner and define what monosaccharides are. Monosaccharides are those simple sugar which cannot be broken down or which cannot be hydrolyzed further. Oligosaccharides, unlike the monosaccharides, these oligosaccharides are made up of 2 or few monosaccharides that are joined together. So in my slide, you see it as monosaccharides plus monosaccharides. The third classification of carbohydrate is the polysaccharides. Look at the name. The word poly is there. The prefix poly is put up there. So poly means many. By this way, we can understand polysaccharides as the carbohydrates that are composed of large number of sugar molecules. Because they are composed of large number of sugar molecules, this also have heavy molecular weight. Further, the monosaccharides are divided into different types based on the number of carbon atoms. We're going to look into this. The first type of monosaccharide is called as the trioses. As is indicated in the name itself, trioses, trio means 3. So this type of monosaccharide possesses 3 carbon atoms in the hydrocarbon chain. Please have a look at the screen. And please count the number of carbon atoms. You will see 3 carbon atoms in the entire hydrocarbon chain there. That's why they're named as trioses. Example is glyceraldehyde. Tetroses is the second type of monosaccharides. It is composed of 4 carbon atoms in the entire hydrocarbon chain. You may count the number of carbon atoms there as well. You see 4 carbon atoms making up the tetroses. Example is erythrose. Ribose is the next type of monosaccharide type. It is called as pentoses because if you look at the number of carbon atoms there, it's going to be 5. We are also going to touch on ribose in the later slides. Hexoses like glucose are the everyday carbohydrates that we come across. We even include them in our diet or drinks. And then if you look at the number of carbon atoms in the hexoses, you would find 6 numbers of carbon atoms in the hydrocarbon chain. The example is glucose. The last type of monosaccharide is called as the haptoses. Haptoses because it contains 7 numbers of carbon atoms. An example is pseudoheptulose. In this slide, let's look into the types of oligosaccharides. As defined earlier, you may recall that oligosaccharides are the sugars or carbohydrates that are composed of two or few monosaccharide molecules. Oligosaccharides are further classified as disaccharides, trisaccharides, tetrasaccharides, pentasaccharides, and hexosaccharides. But our scope of discussion is going to only revolve around what is called as the disaccharides. So disaccharides are composed, or these are the sugars which are composed of two units of monosaccharides. Let's look into few of the examples of disaccharides. We have sucrose, maltose, and lactose under disaccharides. Now, this sucrose and maltose are found only in the plant cells. Sucrose and maltose are only found in the plant cells. While on the other hand, lactose is found in the animal cells, which is why they also call as the milk sugar. Please come back to sucrose. They are also called as the cane sugar. Cane sugar because these are adequately found in plants like sugarcane, beets, carrots, and fruits. This is the storage carbohydrate of the plants. Unlike the sucrose, maltose does not occur in the nature. These are found in the germination of starchy seeds and malls. Perhaps that's the reason why maltose are also called as the malt sugar, and these are also used in brewing alcohol. Lactose is found in animal cells as spelled out earlier, and this is the only carbohydrate that is available to the sucklings. By sucklings, I mean to say infants. I hope students viewing this session are getting something out of the biomolecules that I'm talking. Let's look at the polysaccharides type. Based on the functions of the polysaccharides, these are categorized under three headings. The first one is the storage polysaccharides. The second one is the structural polysaccharides. The third one is the mucopolysaccharides. Let's try to see the definition of storage polysaccharides. As name indicates, this polysaccharides serves as the food reserve. So under this, we have starch and glycogen. Let me first talk on the starch. This is the form of carbohydrates that serves as the food reserves in the plant cells. And these are also the end product of photosynthesis, where the green plants, a process where the green plants prepare their own food. As and when the cells need, the starch can be broken down into the subsequent sugar units and can be taken up readily by the cells. Glycogens are the storage polysaccharides that are found only in the animals. In mammals, they are stored in the liver and muscle cells. And these glycogens are released in times of emergency. For instance, you're out on a walk and suddenly you encounter a tiger coming face to face with you. What would you do? Your liver will immediately break down the glycogen into glucose, and that glucose will be poured into the blood and will be distributed across your body. Why does the body come up with this kind of mechanism? Because encountering a tiger is an emergency situation. You'll have to either beat the tiger or you'll have to take a flight. So there's only two options there, fight or flight. So in times like this, the glycogen that is stored in the liver will be broken down into glucose. By the time it's broken down into glucose, it means they can be readily taken up by the cells because you need extra energy. Your heart has to beat faster. So the process of breaking down the glucose, breaking down of glycogen into the glucose is called as the glycogenolysis. Structural polysaccharides like cellulose are the main components of cell wall in the plant cells. They provide for the structural functions of the plant cells. And the third type of polysaccharides is the mucopolysaccharides. They are called as the mucopolysaccharides because they produce gel-like substances called as the mucilage. These are sticky in nature, and then this is the reason why they have these adhesive properties. Alright students, we have talked a lot more about the first molecule of the carbohydrates, and then it's also my hope that you have picked up something from the carbohydrates and its types based on the carbon numbers, based on the sugar units. Now let's move on to the second type of the biomolecule, that is proteins. The word protein is derived from a Greek word proteus, which means primary or holding the first place. This signifies the importance of proteins. Perhaps if you look at your own body, almost all the structures are made up of the proteins. These proteins are the macromolecules or large molecules that are put up of polymers or large subunits of amino acids. Please don't worry about the amino acids. We are going to talk about the amino acids in the next slide. For now, please know that these proteins are composed of polymers of amino acids. Proteins help in the construction of cartilages and connective tissues. They help us fight against antigens or foreign particles by producing antibodies. They help us produce hormones and carry out various biochemical reactions in our body. These are required for structure, function, and regulation of the body's cells and tissues and organs. Amino acids. These are the monomers of proteins. If you have difficulty in getting the idea of monomers, please recall my simple analogy of an individual chains coming together to form the entire chain. The individual chains can be put up as the monomers, while the entire chain can be thought of as the polymers. So these amino acids are the monomers of proteins. These are organic acids composed of carbon, hydrogen, oxygen, and nitrogen. The figure that pops up on the screen shows the molecular structure of amino acids. If you look at the right-hand side, you would see carboxyl group, COOH. If you look at the left-hand side, you see NH2, which is the amino group. And if you look right at the carbon atom in the middle, you will see R, which is the side chain. So this R can be polar, this R can be non-polar. This R can also be amphoteric in nature, meaning they can possess both the properties of acids and base. Before we move on to the next slide, please have a look at the molecular structure of amino acids and then see if you can recognize the different types of molecules that are found on the amino acids. I'm not sure if you have heard of these words. Did you ever think essential amino acids could be essential and non-essential amino acids? Well, if you have not, we are going to discuss it in the lesson. As you can see it on the screen, these amino acids are divided into essential amino acids and non-essential amino acids. I'll be talking mainly about the essential amino acids because we have to know this, and this is quite important when we talk from health point of view as well. Of course, we are not neglecting the non-essential amino acids as well. Firstly, we'll look into the reason why these are called as the essential amino acids. The word essential means something which is indispensable. That means these classes of amino acids are required in our body. But unfortunately, our body cannot synthesize this class of amino acids. That's why these are termed as the essential amino acids. Once again, these are termed as essential amino acids because these are indispensable because they cannot be synthesized by our body. They have to be supplemented in the diet that we consume, like meat or cottage cheese or dairy products. Together, the 10 essential amino acids and 10 non-essential amino acids are called as the magic 20. This term was coined by one of the discoverers of DNA, Francis Crick. He called all these amino acids as the magic 20 because starting from bacteria to man, all these amino acids are used in the synthesis of a protein, which is the reason why Francis Crick termed all these amino acids as the magic 20. In here, we're going to look into some of the functions of amino acids. Of course, there are many, but we are only going to look at some of the pertinent functions. The first function is the protein formation. Like I said in the beginning of the protein lesson, that amino acids come together, forms larger unit called as polymers, and then finally this forms up the protein. Hence, amino acids are the building block of protein formation. Several specific types of amino acids come together and then they give rise to compounds. And these compounds are further converted into various other essential chemicals in our body like hormones. Glucose formation. If you can recall the molecular structure of amino acids, you will see that it contains 3 different groups: the carboxylic group, amino group, and the side chain R. Now what happens is, if you remove the amino group from the molecular structure of amino acid, the remaining structures is going to give rise to glucose. Several non-protein amino acids forms the basic components of antibiotic. So amino acids are also used in making antibiotics. Several amino acids form important components of antibiotics. And they also serve as the biological buffers because of the fact that they possess both basic and acidic properties. This sums up the essence of the proteins. It was quoted by the Nobel laureate Yoshinori Ohsumi. He said life is an equilibrium state between synthesis and degradation of proteins. Lipids are fats and fat-like substances with low solubility. They are made up of carbon, hydrogen, and oxygen. They are, however, soluble in organic solvents like alcohol and benzene. As compared to the energy released by the carbohydrates, the energy released by the lipids is double. So the energy released by the lipids is more. Let's quickly look into the classification of lipids. They're classified as simple lipids, compound lipids, and derived lipids. I'll just give you the definition of each classification of lipids, example, and then move on to the next slide. Simple lipids are those lipids which contains esters of fatty acids with alcohols. Examples include butter, milk, and soybean. Compound lipids are those lipids that contains esters of fatty acids along with carbohydrates and proteins. Example, cephalins. These cephalins play important role in blood clotting as well. Derived lipids, as indicative of the name itself, derived lipids are those lipids that are made up of simple lipids and compound lipids. Example includes steroid hormones like estrogen, female hormones, androgen, the male hormone, and glucocorticoids, the stress hormone. Just like the amino acid is to the protein, fatty acids are for lipids. So quickly, fatty acids form the building blocks of the lipids. We're going to look into 2 types of fatty acid: saturated fatty acids and unsaturated fatty acids. Saturated fatty acid contains double bond between 2 carbon atoms. Examples are butter fat, meat fat. Unsaturated fatty acids contain one or more double bonds. Examples include sunflower oil and mustard oil. With this, we move on to the DNA. DNA is called as the secret of life because it contains the genetic information that has to be transferred from parents to the offsprings. It also acts as the blueprint of our life because the instructions to carry out various life processes has to come from the DNA. It was discovered by Nobel laureates James Watson, Francis Crick, and Maurice Wilkins. Let's try to break down into different components and study this one. Nucleotides, or in other terms, the building blocks of DNA, are composed of nitrogenous bases like adenine and guanine, which makes up the purine, thymine, cytosine, and uracil, which makes up the pyrimidine. And they contain pentose sugar by the name deoxyribose. And the third component of the DNA is the phosphate backbone, which is highlighted in the yellow. Is also marked with the orange marker over here. So this makes up the components of DNA. Let's look into the structure of DNA. This double-stranded helical structures, I'll quickly draw it on the board. These are helical structures intertwined with each other. If I were to unwind the double helical structures, it will look something like a ladder having a small strands in between. These small strands that joins the two strands can be thought of as the nitrogenous bases, and the two longer strands by the sides can be thought of as the helical structures of D and A. So for our easy understanding of the structure of DNA, I have put up a skeletal structure of DNA. The last biomolecule I have in my lesson is the RNA. If you have understood the concept of DNA, or if you have got the idea of the structures and the components of DNA, this is going to be easy as well. So these are synthesized in the nucleus, but are mainly found in the nucleolus and cytoplasm. It helps in the synthesis of proteins. These are some of this RNA serves as the hereditary material of some viruses like coronavirus. Unlike the double-stranded helical structure of DNA, these are single-stranded. Let's have a look at the components of RNA. Nucleotides in the DNA serves as the building blocks of the DNA. In a similar manner, the nucleotides in the RNA is called as the ribonucleotides. Let's look into its components. These are composed by nitrogenous bases, purine and pyrimidine. I would like to have your attention on the screen and make out the difference. In case of DNA, it was thymine, but in case of RNA, it's going to be uracil. So this is one of the main differences between RNA and DNA. And they are composed of sugar. It was deoxyribose in the DNA. It is ribose in the RNA. And similar to the DNA, it also contains the phosphate backbone. Here I present you the structure of RNA. Once again, I would like to remind you, please take note of the nitrogenous basis uracil. Uracil is in case of RNA, whereas it is thymine in case of DNA. Now this is the skeletal structure of RNA for our easier understanding. So in summary, I'd like to flash up a beautiful quote by a Nobel Prize winner in chemistry as well as Nobel Prize winner in peace. He said, "Life results due to a relationship of molecules and is not property of any one molecules." A quick check of what we have studied. I'm going to pose you these three questions. Question 1 says, Sonam is a virologist who is studying on a genetic material of a coronavirus. What is the equivalent term of the genetic material in an elephant? Question 2, sometimes when you're exhausted and try to concentrate on your studies, you feel like you have headache. Which biomolecule do you think is running short in your brain? Question 3, and perhaps the last one, how does genetic material of a dog differ from genetic material of a bacteria. I come to the end of my lesson. I hope you had a good time listening to my lesson. I hope to see you all in my next class. Thank you so much.